All right, it looks like we're live. So I'd like to welcome everybody to this month's micro seminar, the first one post 2016 election. So we made it, we survived for the month at least. Today's presenter is Ameet Pinto. Ameet's got a pretty varied background. So he had his undergrad in chemical engineering at the University of Mumbai, then did a master's at the University of Alaska, so changing climates pretty rapidly there. After his master's, he went and started his PhD at Virginia Tech with Nancy Love, and he said in his last year of the PhD, Nancy moved to University of Michigan. Ameet stayed in Blacksburg, but then realized that there was a postdoc opportunity in another lab in Michigan. So then Ameet postdoced in Luke Raskin's lab at the University of Michigan for a little while, and then in January of 2016, he started his own lab at Northeastern University in Boston. He tells me that he just got his lab keys a couple months ago, and he's going to have a Thanksgiving Day lab setup celebration. So he's at the fun point of his career when flush with money and no worries about committee meetings yet. So with that, I'll leave it to Ameet, and I'll see you guys in a little bit. I can't hear you, Ameet. Are you good? Are you?
Oh, can you hear me now?
Yeah, you're good now.
Okay, that's great. Thank you so much for inviting me to present here. David and I met at the Michigan Microbiome meeting in the spring or early summer, I believe, and we got talking. I've been following Micro Seminar since it started, either watching the talks live or on YouTube when they get posted. So it's really fantastic to be able to present over here. If I can keep my presentation up. I am an environmental engineer with interest in microbial ecology, and a large chunk of my research focuses on drinking water microorganisms. And today I'll give a brief overview of the research that we are doing, research that we want to do in the future, on how do we manage microorganisms at the tap. Before I get on with my talk, a brief plug about my university that I'm at right now. So Northeastern is located in Boston, Massachusetts, and this is sort of looking eastwards. You can see the skyscrapers in the center of town. It's not too far from the center of town. It's a very well-situated campus. And within Northeastern University, I am in the Civil and Environmental Engineering department. We have about 44, 45 core and affiliated faculty. Everybody is research active in areas from environmental engineering to geotech, structures, construction, and transportation. So in terms of research activity in the department, we're fairly diverse and fairly active. So if any grad students out there are considering a PhD or master's in civil and environmental, look us up and get in touch. Our focus, because we are an urban university, our focus, our vision is urban engineering, and our different sort of research themes such as environmental health, sustainable resource engineering, civil infrastructure security. And within the environmental health clusters, I fall into the category of water and ecological systems. So my research group, we like to think of ourselves as researchers who do ecology-driven combination of experimental and computational approaches for water engineering applications. So whether it's ecological theory, whether it's physiological theory, we derive inspiration from that, perform our experimental work, which is a combination of microbiology, some microcosm work, and a lot of field work. And so that's what I'll primarily be talking about, the field work that we've done. And on the computational side, we dabble with bioinformatics, collaborate also quite extensively with bioinformaticians and stochastic modelers, and then incorporate multivariate statistics to see if our experimental data conforms to the sort of theory that inspired those set of experiments. When I speak about water engineering applications, probably about 90% of the work that I do and my group does, we're interested in, is related to drinking water microbial ecology. that is the ecology of microbial communities that inhabit our drinking water systems. And the reason we focus on it is obviously it has broad implications for public health and well-being. All of us consume water every day. We are exposed to microorganisms in our water supply every day. But it also has a significant impact on industry. And I'll revisit this theme as we move through this talk. And finally, microbial communities in drinking water systems are vastly unexplored. And I'll also present some evidence to make that point a little bit later on. So for those of us who are not familiar with drinking water treatment technologies that are out there, most of the treatment technologies that are deployed in water treatment plants, from a biological perspective, they're geared towards minimizing growth substrate available for microbial growth. So once the water is treated, it has to go through a long sort of extensive network of pipes to actually make it to your homes. And water utilities want to make sure that there is no microbial growth in the distribution system after the water is distributed. So to do this, they reduce the growth substrate, as you're seeing with this. This is just a depiction with the blue area. And at the same time, the focus is on increasing the chemical stress. And the types of chemical stress I'm talking about are, for example, some water utilities will use ozonation for disinfection, primary disinfection. A lot of water utilities in the US will use, and actually across the globe will use, chlorine or chloramines for disinfection and maintain a disinfection residual in the distribution system. So it's sort of a combination of very low amount of growth substrate available and a high amount of chemical stress in our water system when the water is actually sent out and makes it to our taps. Now, despite this, what we find is that tap water typically has about 10 to the 3 to 10 to the 5 cells per mil, maybe 10 to the 6 cells per mil. This is some flow cytometry data from Frederick Hamas's group in EWAG, where he puts into perspective what the microbial abundance in tap water looks like in the context of other aquatic ecosystems. So definitely a lot lower than wastewater, but there is still an abundant microbial community. So you're probably at the lower end of the range for tap water over here. If you're looking at disinfected water systems, such as in the United States, but there are countries across the world that do not disinfect their water or maintain no disinfectant residual in their distribution system. So Netherlands is a prime example. Switzerland is also an example. So they don't really maintain a chlorine residual in the distribution system. But despite sort of this very abundant microbial community, one of the things, the research focus of the drinking water sector is primarily driven by one of the core missions of the water utilities, and that's to make sure that people don't get sick after consuming water. So we are looking at a very sort of small overlap between, quote unquote, the drinking water microbiome and the human microbiome. And this disease overlap has driven the research agenda in the drinking water sector for a long time, and rightfully so. Obviously, we don't want pathogens in our drinking water system. So this is just a simple, this slide shows a simple web of knowledge search where I use some key terms and use them as topics on Web of Knowledge to find out how many publications there have been over the last 15 years or so. So, around 2000, maybe a little bit before that, is when molecular tools really started being applied, maybe in the late 90s, when molecular tools really started being applied in drinking water and wastewater research. So over the last 15 years, the drinking water sector, pathogen-focused research has probably published about 1,000 papers a year, about 800 papers a year. But when you think about a community-level focus, what proportion of those studies have looked at microbial community as a whole? Now, whether it's culture-dependent, whether it's culture-independent, the gap is significant. We've not investigated the drinking water microbial community as a whole. And that is essentially this blue section that we perceive does not overlap with the human microbiome. If we look a little bit further, how many of those studies have looked at drinking water microbial communities and tried to include some microbial ecology perspective in there? The number of studies changes quite a bit as well. So there is this big gap between what's in our water supply in terms of the microbial community and what we focused on traditionally. Now, changing that, enhancing the number of studies, getting a deeper perspective into the microbial community or microbial ecology of drinking water is not an easy task. People are rightfully focused on pathogens. They make people ill. So there has to be some incentive for the industry to sort of transition from a pathogen-centric focus to so a community-level focus. So that's one of the challenges. How do we incentivize the drinking water industry to start, okay, look at pathogens as well, but start looking at the community as a whole? And when I think about it, there are sort of, you can put those incentives in several different boxes. So one, community level investigations can help secure public health, and I'll give some examples of this as we move on. But that's not the only impact of microbiology or microbial communities in drinking water systems. Some of these microorganisms are involved in corrosion, microbially mediated corrosion. We lose a lot of water through leaks in our drinking water distribution system, sometimes in our premise plumbing. Microorganisms can also affect the aesthetic quality of water. And on the plus side of things, we can also use microorganisms for biological treatment. So instead of using chemicals to remove pollutants from source waters during drinking water production, we can actually exploit the metabolism of microbial communities to remove those pollutants. So that's a sustainable twist on drinking water treatment. So I'll give you one example of how we can motivate industry to look at community level investigations. And that comes from the perspective of public health. Now, people usually don't get sick from consuming tap water. There is lots of sampling that takes place. There is rigorous treatment that takes place in most places. So tap water is extremely safe for the most part. But how do we make sure that it stays safe during distribution through our pipes? So this is data from UK. It's a couple years old now. That shows the number of samples that were collected in England, Wales, and Scotland. In I think 2013, maybe 2014, I don't remember now. So about 700,000 samples were collected for testing for E. coli. So that's a huge amount of money that's spent collecting samples. People have to go out, collect a sample at various sampling locations, bring the samples back to the lab, then a dedicated technician analyzes those samples. So it's quite an expensive procedure. At the same time, the number of samples in that year that were actually positive for E. coli, so positive for E. coli being the water is not safe to consume, and there needs to be some sort of mitigative approaches, mitigative interventions that need to be established at that point. That were only about 70 samples that were positive. So there is this, we can celebrate to one extent that, hey, 99% or greater than 99% of the samples are compliant with regulations. Fantastic. But we can also look at this picture and ask a different question. That is, does this gap between the number of samples collected and the number of positives, is this a gap between perception and reality? Do we perceive that our water is less safe than it really is? So this difference between perception and reality and the quest for greater than 99% compliance is actually quite expensive. You can imagine the amount of resources, person time, lab resources that are spent collecting all of these samples. And when you sort of layer on the fact that most of the data actually only becomes available about 2 to 3 days after the samples have been collected, that means the water probably was already consumed for those 70 events that were detected to be positive for E. coli. So that's an issue. So we're spending all these resources as an industry, collecting these samples, analyzing them, but at the same time, it's very reactive in a sense. So from my perspective, sort of water quality monitoring, which is very pathogen-centric, is more of a forensic exercise. Yes, we do rigorous treatment at the drinking water treatment plant, supply the water, It meets all the standards, but then we sort of have a wait and see approach to some extent, in that we want to detect the events that happen as early as possible and then try to mitigate it. So one way to incentivize looking at microbial community as a whole might be a new way of doing things, is instead of relying on event detection and mitigation, we could transition towards event prediction and correction. That means maybe there is potential to make observations at the drinking water treatment plant and then predict what the quality of water is going to be at different regions in the distribution system. And the reason doing a community level perspective matters is because pathogen detection are very sporadic and a few events. So it's difficult to collect sufficient amount of data to build this predictive framework. But the community as a whole can be readily detected with the tools that we have at our disposal. So instead of looking for a signal at a very low level of abundance, if we look for a signal at a higher abundance level of microorganisms, maybe we can start transitioning from an event detection to an event prediction and correction framework. And so if we want to try to move towards this approach of event prediction, some of the key factors that we have to think about is, okay, how does treatment process impact the microbial community? How does this microbial community in our water systems change over time? How does it change over space in the distribution system and drinking water treatment plant? And some of the motivation for these questions and that I'm pursuing in my group here, a lot of the motivation came from the work that I did with Lou Troskin when I was a postdoc with her in Ann Arbor, Michigan. So we worked with the Ann Arbor, City of Ann Arbor drinking water utility. So those of you who are in Michigan would know this is Ann Arbor right here. That's where the drinking water treatment plant is located. So we started a sampling campaign where we sampled at about 6 or 7 locations in the drinking water treatment plant, maybe 5 locations, I don't remember now, and about 9 locations in the distribution system. And we sampled in this sampled every month at these 15 or 16 sampling locations for a period of about 15 months. So it was sort of a first look at getting a sense of what's the impact of treatment process, what is the spatial and temporal dynamics of the microbial community in the treatment plant and distribution system. And we had a lot of interesting and novel insights come out of this work. So one of the things that we found was that treatment processes, we know that treatment processes are there to act as a barrier between source and the tap. What these treatment processes were doing is actually significantly impacting the microbial community. So this is just an NMDS plot here with the Bray-Curtis distance metric. And Ann Arbor, Michigan uses a combination of river water and well water as its source water. And when we go through the first few steps of sort of physical chemical treatment, this includes softening, coagulation, flocculation, and sedimentation. What we essentially see is a drastic shift in the bacterial community, in this case, once it has been through those first few steps of treatment. If we now move towards the filter, that's where the water goes. It changes a little bit. If we move to the filter effluent, it actually looks very much like the filter. Disinfection, it still looks very similar to the filter and the filter effluent. And then in the distribution system. So there is this nice little shift in the distribution system. But what becomes evident is the fact that the microbial community in the distribution system and presumably at the tap looks nothing like the source water community that was used for treatment, at least over relevant timescales. And so we published a paper back in 2012 looking at the microbial community, and one of the key conclusions was that the filters at the drinking water treatment plant were playing a role in seeding the distribution system, that is seeding the community, microbial community in the extensive network of pipes that is sort of buried underground. We did some diversity balances to come up with these conclusions, but what you can also do is actually track individual OTUs. So this is just two examples over here. On the y-axis in blue, you have the abundance of an OTU in the distribution system, and the y-axis in orange, you have the abundance of the OTU on the filter. And you can see, even though the quantities in terms of the relative abundance is different, you can see this nice synchronous pattern in terms of when something blooms in the filter, you start seeing its effect even in the distribution system. So that's the first sort of insight that we got from this sampling campaign, is that, yeah, the treatment processes really change things up quite a bit. But at the same time, the filter is where the action is really at in terms of seeding the distribution system. And in fact, about 60 to 70% of what we see at the tap can be traced back directly to the filter at the OTU level. The next interesting insight that we got is when we started looking at the distribution system and the temporal patterns of the microbial communities in it, we started seeing some really nice temporal signal. So the square represents the water leaving the drinking water treatment plant, and the circle is a centroid of all the points in the distribution system. So I'm not putting all the points, about 9 points in distribution, out there to sort of clean this plot up a little bit, and this is just a principal coordinate analysis, again using the Bray-Curtis metric. And as we start moving through time, what becomes quite clear is there is a distinct seasonal pattern in the microbial community, or the bacterial community rather. So you can see that this community itself is shifting over time. And then when we come back about a year later, there are about 3 months in June, July, and August. So those are the bacterial community in the distribution system looks actually very similar to the bacterial community in the distribution system a year ago. So this is a very nice, very interesting observation here. The fact that there is not only a seasonality, but there is also the potential that at this particular drinking water treatment plant, there is reproducibility over annual time scale. So I'll just put all the data up here. You can see summer, fall, winter, spring, and then summer back again. So you're actually cycling back to your original community composition a year later. So that's very interesting. If these seasonal dynamics are conserved and we can get a sort of characteristic time scale over which the community changes, then that's quite handy in terms of trying to predict what it's going to look like in the future. So in addition to the temporal pattern, we also saw a very nice spatial signal. What I mean by very nice is sort of unexpected. What you have over here, this is the drinking water treatment plant and the 9 locations in the distribution system. We went back and mapped, with the help of the Ann Arbor Water Utility, we mapped the distance of each of the sampling locations to the drinking water treatment plant, and also the pipe layout. So the dashed lines indicate the length of each of those pipes, and the hard lines, or the solid lines, indicate the surface area of the pipes. So for the blue section that you have over here, you can see that it is a linear flow path as compared to the yellow and orange sections where there is branching from the distribution system. So we did not see a spatial signal in these 2 sections of the distribution system, but we saw an interesting spatial signal in the linearly connected flow path. So on the y-axis, what you're seeing over here is weighted unifrac distance. And on the x-axis in the top plot is the distance in kilometers that are connect 2 sampling locations. And in the bottom is surface area that connects 2 sampling locations, which includes the drinking water treatment plant and these 3 sampling locations. So because of pairwise comparisons, you have more data points here. And what you see is there is a nice, what appears to be a sort of a linear distance decay. So as the communities, as the sampling points become farther away from each other, they become more dissimilar to each other, which is also a very nice and helpful observation. So if we know what the community looks like at the drinking water treatment plant, we can anticipate, at least for this section, we can anticipate what it might look like, say 5 kilometers down the line or 10 kilometers down the line, and so on. So this, so there is a the filter seeds, there is a temporal pattern, and at least for a section of the distribution system, we can pick up a spatial pattern that's distance decay. The next thing we did is we took this data, the data that we had collected over 15 months, and we wanted to see whether if an OTU became more abundant, if an operational taxonomic unit became more abundant, was it likely to be also more widely distributed? And so we went back and we calculated the frequency of detection of an OTU and its mean relative abundance. So there are lots of data points over here. And essentially what it means is if you collected 10 samples one particular time point and an OTU showed up in all 10 samples, then you're talking about a frequency of detection of 1. And if it showed up in only 2 samples, then you're talking about a frequency of detection of 0.2. So you can see there is a very nice relationship over here between frequency and the mean relative abundance. We then went back to the literature, started digging around in and came across this concept of occupancy abundance relationships, where the occupancy of an organism scales with its abundance at any given location. And one of the models, we tried several different models, and it's in an MBio paper now, a couple of years old. We tried several different models, and one of the models that fit really well for our data was the Hansky-Gillenberg model. This was published in 1997, and the red data shows the model fit. So for the most part, what it seems like is also the fact that an OTU is going to be much more widely distributed across the system if it is also abundant, which is also an helpful observation. So if I step back a little bit, and if this model is calibrated very well, if we have confidence intervals both along the y and the x-axis for something like this, then we might be able to use this data to, say, measure the abundance of an OTU at a few select locations, come back to this model fit, and then try to estimate how widely it's going to be distributed across the locations that were used to calibrate this particular model. So from those studies, it was quite nice that we had these 4 themes that emerged from that spatial temporal sampling. And the 4 themes were, okay, the filter seeds, that's fantastic. We're seeing some signal of distance decay. We're also seeing some seasonal dynamics, some annual reproducibility. And the frequency of detection of an OTU is a function of the abundance of the OTU as well. So how do we make this useful? We have these observations. Can we just take them and run with it? As it turns out, one of the things that we did is then start asking questions. Okay, we have 4 different things that we can work to get together, but maybe we should step back and ask some very basic questions. First, are we sampling correctly? Because if we want to invest a lot of time and resource in getting huge amount of spatial temporal data, then we should really make sure that our sampling protocol is the best that we can do. Second, are we getting useful information from the type of sampling and data processing and sequencing that we're doing? And if we were to tie these 4 things together into a, say, predictive recipe, that means if we somehow manage to pull all these different threads together to put a framework that predicts what the microbial community is going to look like in a drinking water system in the future, is this going to be generally applicable, or do we have to come up with a custom solution for each utility? And then finally, maybe a little bit more pie in the sky question was, okay, if we can observe accurately, if we can predict confidently, then maybe this opens the road for also manipulating the microbial community. So as engineers, any sort of process intervention should be informed by robust models. So I'll go through each of these questions and how we've tried to tackle them, or how we've fallen short over the last couple of years in trying to tackle them, and the lessons that we've learned from it. So first question, are we sampling correctly? So about the same time that I started asking this question, I moved to the University of Glasgow, where I was fortunate enough to work with Bill Sloan and Joanna Schroeder at the University of Glasgow, both statisticians and deep thinkers. So I started having conversations with them. And after a little time, Bill Sloan, who developed some of the neutral modeling approaches for microbial communities. Um, um, he, he he came up with the conclusion that sampling is non-trivial. For those of you who know Bill, if you put that phrase through a Sloan translator, it translates to your sampling approach is shite. So then we had to step back and think, hey, how can we improve our sampling? And what I mean by sampling over here is not sort of the filtering process of harvesting the microbial cell, maybe not even the DNA extraction process, but just the process of collecting water. Are we doing that correctly? And so this is how we sample currently. We go to any particular sampling location. There is a pipe underground through which water is being transported. We find a tap close by. We flush the tap to make sure there is no stagnant water in the premises plumbing or the building plumbing. And then we start filtering our water. So what's happening over here is our sampling time is fixed in space. But the microbial community that's in the pipe is actually moving through the pipes itself. And so it is moving through space and time. So essentially, by being stagnant in space, what we are doing is we are getting composite of snapshots of a microbial community as it moves through the pipe. We aren't really capturing what happens to a packet of water or a volume of water as it moves through the pipe. We are getting a composite snapshot of multiple packets or volumes of water. So some of these discussions led to a very interesting sort of simulation paper that we published in PLOS One last year, where we tried to simulate sort of what happens to the packet of water if we track the same packet of water as it moves through the pipe in terms of the microbial community. But that was just a simulation exercise, a very informative and interesting simulation exercise. How do we translate that into reality? Because if we want to really understand the mechanisms that are governing how microbial community is changing in the drinking water distribution system, we ought to try to understand some of the things like biofilm detachment, growth, transport, decay of microbial cells. But this composite of snapshot sampling approach doesn't let us do that. What we ideally need is sort of a Lagrangian framework. So our previous framework, even the framework we're using right now, where we are fixed in space, is the Eulerian framework of sampling. What we sort of need is a Lagrangian framework of sampling, where we are following the water as it moves through the pipe. And we were certainly not the first people to think about this. This has been going on now for several years. So this paper back in 2012 from Ed DeLong's group, what they did essentially was collect samples beneath the free drifting surface float. So samples were being collected as the float moved with the current, and sort of they've used the sampling approach now in several different papers subsequently. And it's quite fantastic the kind of insights you can get in terms of microbial interaction and population dynamics if you can sample that way. But in drinking water systems, it's really challenging to do that. First of all, these pipes are under high pressure. These pipes are of varying diameters, and they're buried underground. So how to accomplish this Lagrangian framework of sampling is something that we're still thinking about. It's complicated, but maybe someday we'll get there. So since we can't do the ideal way, let's at least do things reproducibly. And so here is where I started working with Melina Batista, a PhD student, and Svetlana, who was a visiting researcher from Serbia, who visited Glasgow. And we started talking about how do we sample reproducibly. Let's at least, the things that we can do, let's try to do them well. And so one of the things that we wanted to figure out was if we picked a sampling location and made it our long-term sort of sampling location, then we ought to figure out, does the microbial community change throughout the day? Water use patterns change throughout the day, the stagnation time changes. So are we seeing some significant shifts depending on time of day of sampling? If we collected multiple samples, are we going to see the same thing? And if we have replicate libraries, are we going to see the same thing? So that is one sampling campaign we did in 5 houses. It resulted in close to about, with the levels of replication, it resulted about 270 sequencing libraries. And then the second sampling campaign, we wanted to see, does the flow rate at which we open the tap, does that matter, right? So you can imagine if the flow rate is very low, and then you transition to a high flow rate, suddenly you have turbulence in your pipe system, and that has the potential to detach some biofilms. So does the flow rate of the tap matter? And does the sample volume matter? If we collected 1 liter of sample versus 20 liter of sample, are we going to start seeing more and more microbial diversity? So these were the 2 sampling campaigns that we did. One of them we've published, sampling campaign 1. The second one, we're still working on the manuscript. Sorry, apologies to Svetlana and Melina for delaying that. I won't go into all the details of the results, but just the sort of broad conclusions that we came out with. One of the things that we found out was, if you looked at the microbial community, both in terms of structure and in terms of membership, there was a change over the period of the day. So on the y-x-axis, you have sampling time period. This is from 8 in the morning to noon to 4 p.m., and then ending from 4 in the morning till 8 a.m. And on the y-axis, you have chow index. We saw very similar trends, irrespective of whether it was chow index or whether it was observed OTUs. The exact numbers changed though. But what we realized is, yeah, okay, we're going to see changes throughout the day in terms of the microbial community that we are detecting. So if you pick a sampling location, what we have to do is make sure that we sample at that same location at the same time point for the duration of a long-term sampling campaign, just so that we try to take out the variability in terms of what the effect of water use patterns on the microbial community at that particular location. So keep the sampling time point consistent was one of the key conclusions. The second conclusion that we came out with, because we did sampling replication and PCR and sequencing replication, is that below a relative abundance of about 0.01, sampling replication became quite essential. So OTUs that were less abundant than that, we would see them in one sample but not see them in another, and so on. So you need lots more sampling replication. And this is for a system where you're talking about 10 to the 3 to 10 to the 5 cells per mil. But above 0.01% in terms of relative abundance, you need PCR and sequencing replication to make sure that the relative abundance at which you're capturing those OTUs is consistent. I don't have a good answer for how much sampling replication you need for the lower abundance OTUs. But for now, what we're doing is we are doing a lot of PCR and sequencing replications, and being clear about the fact is that we start moving down the abundance scale, our observations become less and less certain. And the reason we are not doing sampling replications is because we use filters, and each filter costs about $5 a pop. And so suddenly, if you start doing lots and lots of samples, the cost starts skyrocketing. So for now, we're focusing on PCR and sequencing replication. In the future, potentially, we'll start adding sampling replications once we figure out how many samples at the same time point we need. And the last conclusion that came from it is, on the y-axis, what you have is the DNA yield. On the x-axis, what you have is either volume of water filtered or the flow rate at which the water was filtered. And what you see, obviously, as you start increasing the volume of water filtered, the amount of DNA goes up, and the shapes are based on the locations that were sampled. And in fact, we didn't observe any effect of flow rate. So these were lower volume samples, these were higher volume samples, and there is no statistical difference in terms of the flow rate of the tap that we were sampling at. If we go and take a look at the microbial community itself, also, we don't see a significant effect of volume and flow rate on the microbial community. But what the recommendation that we came up with is that, collect enough volume of water to where you can get DNA that is significantly above the background contamination level. So when we do qPCR on our blanks and our sort of extraction blanks, or even the filter blanks that we also extract from, we always see something like about 10 to the 2 copies of 16S per microliter of DNA extract, but we try to keep our sample qPCR quantities, say 10 to the 4, 10 to the 5, maybe 10 to the 6 copies per microliter. So that's the only recommendation in terms of volume of sampling, and keep the flow rate constant. Okay, so are we sampling correctly? It's not ideal, but we're keeping things consistent at the same time, thinking about how we can implement a Lagrangian framework for sampling. The next question we wanted to ask was, are we getting useful information? So we've primarily been using 16S rRNA gene sequencing approach. It's obviously very powerful, scalable, extremely useful, but I think most of us also know some of the limitations of it. Short fragment, you can get an OTU, often not, sometimes not get good taxonomic resolution and stuff like that. So, but 16S is a really helpful tool. What sort of changed my mind is what happened over sort of the last year or so. Actually, in 2015, we were also doing a side project where we done for a different question that we were interested in with respect to the microbial community and drinking water systems. We started doing some metagenomics, and this was in collaboration with Lou Traskin and Greg Dick at the University of Michigan. So we sequenced, did shotgun DNA sequencing, and we extracted a whole bunch of metagenome assembled genomes, or MAGs, from this data set. And you can see them dotted on this phylogenetic tree in squares and circles all throughout. And then we came across this genome, which was, or a genome bin, metagenome bin, which was a nitrospira. Now, I did my PhD in wastewater engineering, and particularly looking at nitrifiers. So when you see nitrospira, you say to yourself, nitride oxidizing bacteria. And when we looked at this genome bin, yes, it was a lineage 2 nitrospira that was in there. But as we started digging deeper and deeper into the genome bin, we started seeing genes associated with ammonia oxidation. So with the 16S, we would have called it a nitride oxidizing bacteria. But with the genome was telling us that this was something completely different. And so what you have is the arrangement of genes on this one large scaffold where a whole bunch of ammonia oxidation genes were, and this is the ESOM map, and this little thing shows you where exactly the ammonia oxidation genes were in this particular cluster. So just to take a little tangent over here, after about months of fiddling with the data, we said, hey, let's submit this. Greg was very encouraging. He said, yeah, we've done the best that we can do with this metagenomic data. So we wrote a letter to the editor saying, hey, this is probably just it could go into a genome announcement, but we think there is something novel and interesting over here. So we're submitting as an observation. So a few weeks after our paper was submitted, 2 very important papers came out from Mickey Wagner's group and Sebastian Lecker and Mike Yetton's group, which showed that there was a nitrospira out there that can completely oxidize ammonia. So which was really fantastic. Subsequently, maybe a couple weeks later, our paper was out as well. But it was sort of this journey of realizing that what you see by 16S can be so dramatically different when you go down to the genome level. It really started making me question whether 16S itself is sufficient. And the reason I say that is, if I'm going to ask a water utility to make decisions or change their protocols using these new using these molecular approaches, I don't want to get things wrong. Saying that, hey, you have a nitride oxidizer when you have something that can also oxidize ammonia in the drinking water system and say in the wastewater system. So to really get useful information, and when we talk about drinking water, we're talking about all the way from public health to biofiltration to corrosion issues. So what we really need to do is, yes, get 16S data. It's the most accessible and scalable method that's available. But to really make some firm suggestions or recommendations to water utility, you have to complement it with metagenomics. And potentially, if it's significantly important, then you need to go above and beyond and maybe even bring in cultivation into the mix. Are we sampling correctly? Answer to some extent. Are we getting useful information? Answer to some extent. And the next question that we wanted to tackle is, are we, okay, if we come up with a set of observations, if we come up with a model that links these different observations together, is it going to be applicable? Is it going to be useful overall, or are things going to be different for each utility? So we wanted to then see, are there differences between drinking water systems, and what factors govern those differences? So if there are certain select factors that govern differences between different drinking water systems, then we can sort of try to adjust for them and not have to redo a predictive framework or a predictive recipe from utility to utility. So we started off with a meta-analysis, and Melina led this effort, where we took the data sets that were available across in drinking water systems from, I think, 7 different countries, and we tried to work with them. These were all 16S data sets, and we came up with some reasonable observations, some that confirmed what we already knew, but like all meta-analysis studies, I think we struggle with the fact that the data the data heterogeneity was quite confounding. So we had many samples from chlorinated systems, CHL, chloraminated systems, and disinfectant residual free systems. But these samples were sequenced on different platforms: 454, MySeq, Ion Torrent, and they also targeted a lot of different variable regions. And so for this study, we largely focused on a reference-based approach, where we mapped the sequences to the Silva database and then analyze the data from there. But we still don't have a clear, concrete answer as how different are things across different utilities. And given the fact that we've also sort of now decided that, okay, 16S is good, but we need to complement that with metagenomics, over the last year, we went, over the last year and a half maybe, we did extensive survey. Maria here led the effort, Maria Saviano. She surveyed across several cities in the UK. Somehow she managed to pack everything she needs into a suitcase, including some sensors for water quality analysis, all the kit that's required for filtering of samples, and planning all the logistics, trying to ship the samples back on, I think, in coolers back to Glasgow. So kudos to her for traveling on the train across the UK. But then with the help of Paul van der Wielen at KWR, we also sampled at about 5 different places in Netherlands. So in each different location, we've sampled each city, we've done multiple sampling locations to figure out what is the differences within a city and what is the differences across cities. And we can infer some of the, and we also did rather lots of water quality analysis. So we have an idea for the carbon concentration is nitrogen, phosphorus, chlorine, chloramine, pH, and so on. So there is several people from my research group involved in this effort, but we're also collaborating with some very, very talented and leaning heavily on bioinformaticians like Umar and Meren, who are helping us with this data analysis. So this is these samples have been sequenced on Illumina HiSeq and using shotgun metagenomics, and this work is still ongoing. So we hope to wrap this up sometime in 2017 and have this out. So I think this will give us a really nice understanding of how conserved a drinking water community can be across different drinking water systems, both when it's disinfected, such as it is in the UK. We have samples that have chlorine, samples that have chloramine as residual disinfectant, and in systems where there is no residual disinfectant, such as in the Netherlands. So hope to have some exciting work on this coming out next year. All right. So then I'll tackle the last question. I guess the second last question, we'll find out whether how custom whatever predictive framework we come up with has to be for each utility. But let's, as I wrap up the presentation, I'll briefly look at my last question here, is if we can predict, can we manipulate? Like I said, I did my PhD in wastewater engineering. Wastewater engineers know very well how to manipulate the function of biological reactors. They have a lot of design variables that they control, such as solids retention time, hydraulic retention time, sometimes the electron acceptor, sometimes supplement electron donors. So, but can we do this in the drinking water side? Most of the microbial community in drinking waters happens to be not engineered. It is whatever survives the process. So about a couple years, now 4 years ago, Lute and I, we published a paper in Environmental Science and Technology, which was talked about filtration processes, seeding the distribution system. And towards the end of the paper, we made some very small suggestions. It was a 7,000-word paper. There was a comment in there that says, hey, maybe we can engineer this to be beneficial to human health. So these 7 words seem to dominate the conversation, and I think often got significant and maybe overblown attention. We don't know if we can control microbial communities. We don't know what the endpoint would be if we can control microbial communities, but we have some hints. So we don't know what the endpoint should be for sure, but we have some hints in how we can potentially control it, right? We don't know if it's going to be successful, but it's at least possible. And that's because we know that, across the systems that we've studied, and several other groups now have studied, we've shown that filtration processes have a significant influence in the microbial community. In fact, filtration seeding is a dominant mechanism for how the community in the distribution system assembles. So then potentially maybe it becomes a design problem. How do we design and operate these filters to have the community that we want in our filtration systems? Now, what kind of community we might want in our filtration system is an open question. I don't have an answer to that, but at least we can start thinking along those lines of engineering the microbial community in the distribution system rather than just letting it occur the way it is. Anything we do, any efforts to manage or engineer the microbial community has to be extremely well tailored and needs to absolutely sort of weigh the benefits and the risks. So we are talking about drinking water over here. This is not something we should obviously take very lightly. But if we can observe something well, if you're confident about our observations, if we can predict how those observations are going to change over space and time, spatially and temporally, then at least at the lab level, we can start tinkering with how do we manipulate the community. So modeling community dynamics is absolutely critical to even start thinking about interventions from a biological perspective. And with that, I'm going to thank my research group, which is right now distributed over Glasgow. You see them over Skype over there and over Northeastern, and the various funding agencies that were generous in supporting my research when I was at the University of Glasgow. With that, thank you so much for your attention, and I will stop sharing my screen and take questions.
Thanks a lot, man. That was good. I have one actually to start off, and it's a process question. So I know, so there's designated protocols for monitoring bad microbes in the water system, but when you're just sampling and you see 16S of E. coli or something like that, where it's not according to the designated tests, but you see it, like what do you have to do there? Do you have to report it to anybody, or is it just like, oh, it's
so when we do it, no, we don't report it. There are still a lot of caveats there. DNA, we don't know if it's come from something that's alive or dead. So that's another huge hurdle that we have to get over. So right now, when we find these observations, we will make it to publication. But it's not bulletproof, obviously, any observations. Yeah.
All right. If anybody on the chat has questions or anybody through Twitter wants to ask, I'm looking through now. You guys will occasionally hear a little baby in the background. I'm multitasking right now, is showing that you multitask too. So I'm rocking with my foot, but I'm looking through questions. No questions through Twitter. All right, so actually, I will give you more questions then, because I'm curious. So when you're actually looking at metagenomics of water systems, like what kind of samples do you guys have to do to get good genomes? Because I know it's Now, depending on how many microbes are there, it's going to, you know, what the level of coverage matters, and you know, fragment length matters and stuff. So how do you actually go about getting pretty good metagenomes through these water systems, even if the DNA content is pretty small?
Yeah. So that's a great question. So one, we filter lots of water, make sure we have sufficient amount of DNA to start off with. So our protocol has been right now is we've used the HiSeq in rapid run mode 2x250 right now, and that's significantly improved our assembly. So the latest assembly that we're seeing is much better than anything we did before. So the 2x250 helps. We use the IDBA UD assembler for that to do the assembly. In terms of coverage, you know, I think that's still, I don't have a good answer for that. I think, you know, if it's 10x coverage, then I'd be for any particular metagenome assembled bin, then I'd be quite happy with that. But anything below that is always a little bit suspicious. But you know, I can tell you, we did about 55, 60 samples recently, and on 4 lanes of HiSeq, and nearly 80% of our reads map to the assembled contigs. So our assembly turned out quite well this time around.
You see any of like the I'm blanking on the terminology, but the Banfield lab weird guys that have weird metabolism that they see in the deep ground? Do you see any of that in the tap water, or is that?
Oh, the CPR, or is it? Yeah. No, I don't know yet. I don't know yet. So I'll tell you one example. So when the first time around we did this, when we did 16S, we used to see a lot of cyanobacteria in drinking water, apparently. And now when we did our genome assemblies, they turned out to be Melanibacter. So these are close siblings of cyanobacter, but from the genome, we know these are not cyanobacter anymore. But these are, so again, this is something relates to what Banfield lab has found out before. So yeah, these are, I think we'll start seeing some very interesting things. I don't know if we see any of those unusual bacteria there, but maybe next year I'll have a better answer.
All right, any other questions? Any other questions? Well, if not, I'm just going to thank Ameet one more time. That was awesome. Every one of these seminars is completely out of what I do, so I learned so much from it. So thank you very much. And we'll be back one more in December. We've got Trina McMahon from Wisconsin giving a seminar in December. So with that, we'll see you guys later.